Hi, good evening.
Good evening, sir.
Yes. The others are coming, right? Hello.
Yes, sir.
The other two students are coming. Hmm?
Yes, sir.
Hi, Jishnu. Good evening. one more person is coming up. We'll wait for one more minute, or shall we start? So fine. So what did we discuss in yesterday's the previous class? We started heredity and evolution. Hello, students. Hello.
Yes, sir.
We started right heredity and evolution.
Yes, sir.
Correct. So third chapter only we are going to discuss today. I think we discussed about some of the terms, and let me share my screen. You just check whether you can able to see the presentation. Hello, students. Can you see my screen?
Yes, sir.
Am I audible enough?
Yes, sir.
Yes. So today we are going to discuss about the Grahor Mendel's experiments on pea plants. Like can you say he is Grahor Mendel and an Austrian monk? Look at that. So this is the garden where the major breakthrough in genetics occurred. In this place only, Grahor Mendel carried out all his scientific experiments and discovered the principles of heredity. Okay, very simple garden. Like heredity, genetics. What is genetics first? It is the study of principles of inheritance and variation. Okay, genetics is the study of principles of inheritance and variation. So, what is inheritance? Transfer of characters from parents to progeny. That process is called what? Inheritance. Let me repeat. Transfer of characters or traits from parents to progeny. That process is called inheritance. Variation is when the characters are transferred from parents to progeny because of some endogenous factors or exogenous factor, means the factors within the system or the factors from outside makes a kind of alteration in the DNA that results in variation. So, inheritance, transfer of characters from parents to progeny. Variation is the degree in which the progeny differs from its parents. That is called variation. Okay, so genetics is the study of inheritance and variation. So in this chapter we will be studying that only. And in genetics, why we should study about Graham Mendel's hybridization experiment. Can someone tell me why we should study about Graham Mendel's hybridization experiments in genetics? Yes, tell me. Hello? Sai Vignesh.
Don't know, sir.
Don't know. Okay, fine. See, listen, there are three divisions of genetics: classical or transmission genetics, molecular genetics, and population genetics. In simple, these are all the three divisions of genetics. In classical genetics, we will be discussing about the history and basic principles. Okay, in classical genetics, we will be discussing about history and basic principles. In simple, how genetic principles are understood, the mechanisms of genetics are understood with traditional, conventional, simple experiments. That is classical genetics. Molecular genetics is study about the molecules of genetics like DNA, RNA, protein. Molecular genetics. Population genetics is like what characters are there in me. We are human population. Among individuals of human populations, what are all the characters are common? Within a population, what characters are different? And about all those things we will be discussing in population genetics. So, classical genetics helps us to understand the basic principles of basic mechanisms involved in genetics. So, in classical genetics, Mendel's experiments are the major breakthroughs. Okay, Mendel's hybridization experiments only helped us to understand the pattern of inheritance from parents to progeny. Inheritance is what? Characters inherited from parents to progeny. That rules, what rules they follow, what mechanism is behind that, that can be understood from Mendel's experiment. That is why whenever we study about genetics, inheritance, we start with Mendelian experiments. Hence, also, Graham Mendel is called what? The father of genetics. Who is the father of genetics? Who is the father of genetics? Hello, students?
Mendel.
Graham Mendel. Okay. Why he called us the father of genetics? Because his classical hybridization experiments helps us to understand the patterns of inheritance. Okay, how characters are transferred, that pattern of inheritance we understood with his classical experiments, right? Now, see, Graham Mendel worked on a plant called the pea plant or Pisum sativum, and the important characteristics helped Mendel to describe the pattern of inheritance or the following. Pea plant flowers are bisexual flowers. Means in a single flower there is the presence of androecium and gynoecium components. both male and female components are present in the same flower, and the synchronization occurs. Synchronization in the sense, do you remember gametogenesis of plants? What all the process? Microsporogenesis and megasporogenesis. Where microsporogenesis occur? Tell me where microsporogenesis and megasporogenesis occurs in plants. Hello, students. Microsporogenesis, where it occurs? I think we discussed about sexual reproduction in flowering plants. What are the two components of androecium? Yes. What are the two components of androecium or stamen? Stamen comprises of what?
Anther and
Anther and
anther
filament.
filament.
Very good. Anther
Sir,
is minute.
microsporogenesis occurs in answer.
Yes, anther. That microsporangium. Correct. So, here, that's the main character. In bisexual flowers, like the pea plants are having bisexual flowers. That's the first thing. Second, easy pollination process. Like manually, self-pollination, cross-pollination, prevention of pollination, all these processes are easy with this plant. Okay, so that is another important process. And then, more importantly, the contrasting characters. Nina. Contrasting characters, like all the characters, we are going to discuss this, like Graham Mendel selected 7 characters in pea plants, so all are very contrasting to each other, means two opposite character. For example, tall versus short, round versus wrinkled, pink or sorry purple versus white. So like that, two characters studied by Graham Mendel or all are contrasting. So that's another important thing. And then within 3 weeks, 3 to 4 weeks, the entire life cycle of the plant is completed. Okay, the germination can occur, the growth and development can occur, the fruiting and flowering can occur, seeds can be produced. Okay, almost of on variety. So very short span of time, all the stages of growth can be studied, and they are visible to naked eye also. You can see all the developmental aspects. And then, like here you saw, in little garden the plants were cultivated. So little water, little light, little fertilizers are enough for maintaining the plant. So these are all the reasons, and these are all the common characteristics, like it is a bisexual flower, easy self and cross pollination, the characters are very visible and they are contrasting, and you can see all the developmental stages in few weeks, and then it can be easily maintained. So hence Graham Mendel selected this plant and succeeded in his experiments. Now look at that. So these are all the developmental stages which I said here. Okay, easy developmental stages. Look at that. From the seed, the germination can be seen, and then growth, germination followed by growth of the plant, and then pollination, flowering, pollination, fertilization, and dispersal, all these things again. So easily you can see from the seed germination to again production of the seed, all of the stages of growth are easily absorbed. So that's another important character of this thing. Now Self-pollination and cross-pollination, as we said, they are easy, and it is a bisexual flower. Can you see? Both androecium, see the filament and anther, means this is the filament and top you have anther, and then the carpel called the stigma style ovary is present in the same flower, hence we call them as bisexual flower. And moreover, synchronization, same time maturation of anthers means pollen grain microsporogenesis here, and then here megasporogenesis. Both the time will synchronize also, so at the same time they can occur. So in self-pollination, what can be easily done? Transfer of pollen grain to stigma. Self-pollination, same plant, same flower, pollen grain to stigma. Very easy self-pollination process. And it is a bisexual flower. For cross-pollination to occur, two processes has to be done called emasculation and bagging. Okay, for cross-pollination, two processes can be done manually called emasculation and bagging. Emasculation means cutting and removal of anther from the floral components. Okay, emasculation means what? Anther is cut and removed. Bagging means like anther is removed, and then to prevent open sperm entry or pollen grain entry, they are covered, and then only the desired, okay, the desired, the pollens, see, look at that, here the pollen grains are removed, the anther with pollen grain, the filament and anther are removed, only stigma is left, the stigma style and ovary carpel is left. This can be covered, okay, so this can be covered called bagging process, and with little paint brushes and all, see, this from the stamen of another flower, the desired flower, the pollen grains are taken with the help of simple paint brushes, and they are added to the stigma. So in cross pollination, an artificial manual cross pollination, this can be done. Okay, in the plant, simply self pollination can be achieved. Even we can do that, or it can occur naturally, or also by emasculation, removal of pollen grains, means the whole filament and anther, and then transfer of pollen grains. So this is called what? Cross pollination. So both can be done. Now, like this, I said 7 contrasting characters of Graham Mendel. What are the 7 contrasting characters? So look at seed shape, seed color, pod shape, pod color, flower position, plant order, stem height or stem size. So these are all the 7 character, and 2 contrasting characters are there in all of these 7 characteristics. 2 contrasting characters are there. Look at that. One is round. This is seed shape. Either the seed shape is round, or at the extreme end they are wrinkled. In seed color, yellow seeds are available, or else green seeds are available. Pod shape, inflated, constricted, means full. You can see this is the pod which covers the seeds. Okay, so this is the pod which covers the seeds. And look at the shape of the pod. It is full. Okay, no constrictions are present. It is plain and full. But here, it is not plain. You can see the constrictions all over the pod. So these are all the 2 types of pod shapes available. And then look at the color of the pod. It is green or it is yellow. See, look at the color here. Coming to the seed color, yellow, green. Coming to the pot color, green, yellow. Okay, contrasting thing. And then the flower color. Flowers are purple or else white. Purple and white. And then flowering position. See, look at that, axial, terminal. Axial in the sense you can see the flower on all the axes, in the middle of the rib or in the corner or left side, right side, whatever. So it is present. So this is called what? Axial, along the central axis of the plant. Look at that, this is the central axis of the plant. Here you can see the flowering, and another one is terminal. So only the flowering occurs in the tip of the plant. So this is axial and this is terminal. And then this is very obvious thing, like the stem height is either tall or dwarf. Okay, the stem height is either tall or dwarf. So these are all the 7 characters selected by Graham Mendel. Okay, are you all listening, students?
Yes, sir.
Okay, now can I ask you some simple questions? Yes or no?
Yes, sir.
Yes, sir. Yes, sir.
Okay, so listen, you know what is variation, right?
Yes, sir.
Variation, fine. I will tell you there are 2 types of variation. I will tell the type of variation, but you try to give me the answer. Continuous variation, discontinuous variation. What are the 2 type of variation?
Continuous and discontinuous.
Yeah. And what is continuous variation? Give me one example. What is discontinuous variation? Give me an example.
So continuous variation is where
Hmm?
when the variation helps the organism to survive in the wild. So it gets continuously transferred to the next generation. Whereas non-continuous generation is where it does not help in the life of the organism or hinders its life processes. Then it dies away and does not get passed on to the next generation.
Okay. If it is inherited, it is continuously inherited variation, huh? Or if it is not inherited, it is not, right? Discontinuous. Correct?
Yeah,
In
yes,
that sense you are telling?
something like that. Something like that.
Okay. Fine. See, listen, I will give you another example. Maybe now you can understand. See, listen. Look at the height or color of the skin in your class or in your school or in any, maybe in your family. Maybe it starts with dark color to fair color. Okay, dark color to fair color. In the skin, if more melanin is produced, they are extremely dark. In the skin, less melanin is produced, they are in different color. Or even to understand, you can think about different continents or nations like India or Asia, Africa, okay, Australia, whatever. Look at the ranges in variation in color. See, one person is very dark, very dark. The other person is 90% dark, 10% fair. And another person is 70% dark, 30% okay. And then 50-50. 50% dark, like brown. in India, and then 60% fair, 40% dark, and then 80% fair, 20% dark, and then completely fair. No black pigmentation at all in the body. See here, you can see a range of variation in color. Yes or no? Can you see a range of variation? Yes or no? Color, நம்மளோட
Yes,
color.
sir. Yes, sir. Yes, sir.
Yes. So that is called what? Continuous variation. Continuous variation. Even in your class, some 50 students are there. They may ask in prayer or assembly, they may ask you to stand according to the height. So the first person is 5 feet tall, and then the last person is 6 feet tall. In between, you have one person is 5.1, one person is 5.2, another person is 5.3 feet, another person is 5.4, 5.5, and then finally 6 feet tall. Can you see the height also? Continuously there are some variation. Yes or no? Yes or no?
Yes,
Hello
sir.
students. Yes. So that is continuous variation. But look at here. Are there any differences in between? Either this end yellow or at the other end green. Either full or constricted. Either round or wrinkled. See, there is no in between characters are present. Can you see any of the in between character here? Hello?
Sir.
No. So, such kind of variation is called discontinuous variation. Okay, like either on this end or on that end, no intermediate characters are present. So that is called what? Discontinuous variation. Purida.
Yes, sir.
Okay, so luckily in Grahor Mendel's pea plant, all the characters show discontinuous variation on opposite character. Fine. Now, and you know the basic terms already, like genes, alleles. What is a gene? For example, these both are chromosomes. Here a gene for one phenotype. For example, blood group, you can say this is paternal chromosome, this is maternal chromosome, means this is from appa, this is from amma, father, mother. You can see both are blood grouping gene. Okay, both are blood grouping gene. But appa gave you A blood group gene, amma gave you B blood group gene. Okay, both are blood grouping gene only. Both share same location on chromosomes, different chromosomes, but still one character is based on this or this. The blood group will be, what will be the blood group? A and B in your body, because you get A from, both are dominant genes, you get one appa gene and you get one amma gene, so both are expressed, so you get AB blood group. So to differentiate that, okay, to all are blood grouping gene only, but to differentiate that, A gene gives one color or one nature, like one phenotype, B gives one, means they call allele. Okay, one is called gene and they name it as actually alleles. Okay, two different forms of a gene are called as what? Different forms of a gene. They are called as what? Allele. Different forms of a gene called allele. Can you understand? Hello, students?
Yes, sir.
And like that, genotype, phenotype. See, look at that here. What you see in nature is called what? The phenotype, the visible trait. You can see this stem is tall and this is short. So tall versus dwarf. The visible trait is called phenotype. What you see is called what? Phenotype. The one which determines this is called what? Genotype. Why this plant is tall? Because the genetic composition is like that. Why this plant is short? Because the genetic composition is like that. So we must understand what is phenotype and what is genotype. Phenotype is the visible type. Genotype is the genetic composition which determines the phenotype. And now what you should say know is what is homozygous condition and what is heterozygous condition. Look at here. One, this is paternal chromosome, this is maternal chromosome, upper chromosome, amma chromosome. If both are giving you the same type of allele, for example here also blood grouping gene, for example IA, here also IA, then that is called what? Homozygous condition. May be dominant or recessive. It may be dominant or recessive. Heterozygous in the sense you can see both, for example both are flower color producing genes. One produces purple, one produces white. Okay, one white color producing, the petals color is purple when this yellow gene comes, it is white when green gene comes. So this is called what? If it has two different genes, then it is heterozygous condition. If it is same, it is called homozygous condition. In that also you have variations like homozygous dominant, homozygous recessive. Okay, homozygous dominant and homozygous recessive gene. Now, Graham Mendel carried different type of experiments called monohybrid process, dihybrid process, okay, back cross, test cross. Do you remember those terms? Hello students, different type of crosses. I will tell
Yes,
you
sir.
what are all the different type of crosses. You tell me what they are.
Okay, sir.
Monohybrid cross, dihybrid cross, trihybrid cross, test cross, back cross, reciprocal cross. Okay, at least 6 different types of crosses. What are they? Can you list me again?
Monohybrid cross, dihybrid cross, trihybrid cross.
And then back cross, test cross, reciprocal cross. Okay, fine. Of course, we will be studying about all those things. In simple, let me tell you what are they. But every now and then we will be discussing about those crosses. First, monohybrid cross means cross between, like this cross helps us to understand the inheritance pattern of one gene. Okay, one trait. For example, flower color is one trait. How this flower color is inherited from parents to progeny. When two different type or two same type of parents are crossed, how that single character flower color is there in the progeny. That is monohybrid cross. Dihybrid cross means the parent has two character. For example, round seed and yellow color. Okay, seed shape is round, seed color is yellow. How these characters are inherited to the next progeny. So that is dihybrid cross. For single character, the pattern of inheritance for single character is monohybrid cross. The pattern of inheritance for two characters is dihybrid cross. Okay, and then reciprocal cross in the sense, in the first crossing experiment, male homozygous plant of dominant character and female homozygous plant of recessive character is crossed. Reciprocal in the sense ulta of that. Instead of male dominant, here female dominant. Instead of female recessive, male recessive plant is selected. So that is reciprocal, vice versa. Male versus female, then female versus male. So that is reciprocal cross. Back cross is F1 progeny is crossed with the parent, any of the parent. That is back cross. Okay, what is back cross? F1 progeny. Their progeny itself is crossed with the plant. That is back cross. Test cross means known recessive plant is selected, known recessive character is selected and crossed with unknown plant. Unknown may be of anything. So it may be dominant, it may be recessive, whatever. But known recessive is crossed with unknown. That is test cross. To determine what is the genotype of unknown plant sample. So that is reciprocal cross. Sorry, that is test cross. So monohybrid cross, dihybrid cross, trihybrid cross, reciprocal cross, test cross, back cross, different type of crosses. We will be discussing one after other. Now, coming to the monohybrid cross. What is monohybrid cross? A cross between two pure homozygous parents having single pair of contrasting character. Example, two tall plants were selected. Look at that. One tall, two plants were selected. One is tall, another one is dwarf. Pure plant. What is called pure plant? It is homozygous for any particular character. So that is pure plant. To represent that, we use the term pure plant. So Grover Mendel selected one tall plant and one dwarf plant. Second, she did cross pollination, results in fertilization. So tall plants were selected first step, they were crossed. Tall and dwarf plant were selected first step, they were crossed. That is the second step. Mendel was expecting 50% of tall plant and 50% dwarf plant. But he cannot see when tall and dwarf plant were crossed. He cannot see, he never saw the recessive character dwarf plant. Only he saw all of the plant were tall. So that's the first thing, first step and second step. Now, the result is, in the second step after crossing, all the plants were tall. That's the first step results, F1 progeny. Don't worry, we will be discussing about I'll be showing you the picture also here. Look at that. Two plants are selected and crossed, and all the results plant are showing only one character. For example, tall versus dwarf, but in the progeny only one character is seen, F1 progeny. In the filial one generation, only one character is seen. Okay, only one character is seen. Okay, you can see phenotype and genotype. What is phenotype? What is genotype? Genotype is heterozygous condition. Phenotype is all the plants were tall. Okay, so that's the first thing. Second thing, he crossed all the F1 generation plants. Okay, he crossed it. He selected two plants from the F1 and crossed it. One male and one female. Look at that. This square is actually called what? Punnett square. A scientist named Reginald Punnett helped us to understand or to work out or to predict Mendelian crosses. Okay, the Mendelian crosses. See, now we cannot actually do the plant cultivation and observing the conditions, but we can study. So, to simplify Mendel's work, Reginald Punnett discovered this method. So this square is called what? Punnett square or checkerboard. So step 1 is selection of pure and off plant. Step 2 is like selfing of F1. Step 3 is recording the results, what he got. So this is what is explained here. Step 1, selection of the plant, and step 2, breeding, and then step 3, like in step 3, F2 cross is done, and the characters are interpreted here. This is his first experiment, and he came to know that, look at here, genotype and phenotype. He worked the Mendelian crosses and then listed those things. For example, how many types of genotypes here? 3 types of genotype. One is homozygous for dominant, the first square, and homozygous for recessive, second square, means last square. And then in between there are 2 traits called hybrid traits, means one heterozygous condition, one dominant gene and one recessive gene. So one completely dominant, one completely recessive, and 2 are heterozygous conditions. Okay, heterozygous condition. See, basically what this is experiment, but what we can understand from this experiment? Hello students, so this is cross 1, Mendelian cross 1 experiment, monohybrid cross experiment. What you can understand from that? Means this classical experiment to study or to understand the principles of genetics. What principles can be understood from this simple experiment?
Phenotype is different from genotype.
Hmm?
Phenotype is
Very good. Genotype is heterozygous, one thing. And then okay, fine. See, listen. 2 important laws. Okay, 2 important laws are explained, 2 important principles of genetics, laws of genetics, understood with the help of monohybrid experiment. Okay, 2 important laws. One is called law of dominance, another one is called the law of segregation. Okay, monohybrid cross is connected with 2 important laws. I will tell you what are these 2 important laws. Look at here. When Mendel did his experiments, he just presented, look at that here, when he crossed tall plant versus dwarf plant, all were dwarf, means all were tall, and he repeated this experiments for all 7 characters. Look at that, what are the 7 characters? For example, he selected some seeds with round seed and wrinkled seed. All seeds were round in nature in the first generation. When he crossed yellow seed, yellow color and green color, all seeds were green in color. Like that, even though there are 2 contrasting characters existed, only you can see the top characters. Basically, they are the dominant characters. Okay, dominant characters. They only express in the F1 generation. So, what Graham Mendel means, what we can understand, see, not Graham Mendel did not say anything, means dominance and recessive. He said, like in organism, one character is dominant, one gene is recessive. Okay, one gene is dominant gene. Every organism is having two genes for one character. Okay, two alleles, one from appa, one from amma. Every sexually reproducing organism carries two alleles for one character, one from appa, one from amma. And in that, one is dominant, another one is recessive. Okay, one is dominant and another one is recessive. Here, like when tall and dwarf plant were crossed, why all the plants are tall? He said because tall is dominant character. So that's why all the plant were expressed as what? Tall plant. Okay, so first law of genetics is characters are determined by factors. Factors occurs in pairs. In that, one is dominant, another one is recessive. The dominant gene always has the tendency to express. The recessive gene expresses only in the absence of dominant gene. Note down, recessive gene expresses itself only in the absence of dominant gene. Otherwise, in presence of dominant gene, the recessive gene will not get expressed. That is the first thing. Nowadays, what we call, Mendel did not call, what we call it is law of dominance. Okay, it is called what? Law of dominance. And then another important thing we can notice from this is, see, this is one simple representation, and again this is one simple representation. You can see, even though all the plants are tall here, the genotype, when you look into the genotype, tall also here and dwarf gene also here, and here also you just count how many. For example, I am telling you there are 8 alleles are there. 8 alleles. What are the 8 alleles? Number of 8 alleles. Tell me. How many dominant allele? How many recessive allele? 4 dominant allele and 4 recessive allele. Are you all listening? See, look at that. 1 dominant allele, 2, 3, 4. Capital T 4. And small t 4. You can see 1, 2, 3, 4. And small t also 4. Means here law of segregation is understood. Like the parent gene produces gametes. Okay, gametes are produced. Dominant gene is there. Like that here, parent gene, parent organism produces gamete. Here the recessive gene is also present, and they are inherited together. You can say they are inherited. And here also, if you look into the number 4 tall gene, 4 recessive gene, it represents actually the law of segregation. It explains us the law of segregation. Law of segregation means, no, don't understand clearly. Law of segregation means every character, like there are 2 alleles in one cell, the cells separates by meiosis process. Means in meiosis, 2 set of chromosome from a diploid cell is divided into 2 single sets, like 46 chromosome divides into 23. And the important thing is how the dominant genes are separated and inherited to the next generation. Similarly, the recessive gene also separated and inherited to the next generation. For example, look at here. How many times the dominant gene inherited? 1, 2, 3, 4. How many times the recessive genes are there? 1, 2, 3, 4. So maybe there is a change in the characters like tall or dwarf, change in the phenotypic expression, but the way the dominant gene and recessive gene are inherited is common. That's the most important thing. That is called what? Law of segregation. Okay? In monohybrid cross of Graham-Mendel, we can understand two important laws, rules of genetics. One is law of dominance, another one is law of segregation. Now, please go through these two laws and we will continue then. Just read about these two laws. Hello students, are you all reading this?
Yes, sir.
So, what the law of dominance says, and first, what the law of dominance says?
In law of dominance, it states that both the traits are present in the human body, but only one trait is reflected.
Okay. Then, then, law of segregation. What the law of segregation says? What the law of segregation says? Jishnu, what the law of segregation says? You can't understand. So let me see. Look into the chart. Okay. இங்க பாருங்க இந்த chart பாருங்க. Based on monohybrid cross, two things we can understand. One is the law of dominance. In a diploid sexually reproducing organism, for every character, it may be blood grouping, it may be skin color, it may be any of other character, we have two genes. To determine one character, we get one gene from appa and one gene from mama. We get one gene from appa, one gene from mama. Okay, so in that, one gene is dominant, another gene is recessive. This is what Graham-Mendel is telling. Two genes for one character, and in that one gene is dominant, another one is recessive. What is dominant? What is recessive? The character which is expressed in the F1 generation. F1 generationல இப்ப ரெண்டு plant select பண்ணி cross பண்ணும்போது F1 generationல என்ன character express ஆகுதோ அதுதான் dominant character. அப்போ express ஆகாம இருக்கிற character என்ன அப்படின்னு சொன்னா recessive character. So dominant character and recessive character இருக்கு. இத in simple words எப்படி கொடுக்கலாம்னு சொன்னா characters are determined by factors. That's the first point. In law of dominance, simplify பண்ணணும்னு சொன்னா characters are determined by factors These factors exist in pairs. The factor expressed in the F1 generation is dominant factor. The factor which is expressed in the F2 generation is actually recessive factor. Means the factor which is not expressed here but expressed here in the F2 generation, that is recessive factor. So that's the first thing law of dominance says. Let me repeat, then you have to tell me. First is characters are determined by factors. Factors are nothing but genes. Okay, characters are determined by factors. The factors exist in pair. In that, one is dominant, another one is recessive. What is dominant? The character expressed in the F1 is dominant. The characters which is not expressed here and expressed in the F2 generation, that is recessive character. This is law of dominance. Now, coming to law of segregation, it may be a dominant gene. See, in this condition you can see a dominant gene or recessive gene and a recessive gene. In meiosis, the diploid parent cell produces haploid daughter cells. Okay, in meiosis, the diploid parent cell produces haploid daughter cells. Can you see? Segregation occurs between the genes. One genes goes to one gamete, another gene goes to another gamete. No matter it is a dominant gene, no matter it is a recessive gene, but one gene goes to one gamete, another gene goes to another gamete. Means there is a segregation of dominant and recessive genes during the cell division process. It occurs. So this is called what? Law of segregation. Okay, so that's what is given here. Law of dominance, law of segregation. Okay, see, when Graham Mendel proposed this, okay, when actually Graham Mendel, I'll show you Graham Mendel's paper. When Graham Mendel proposed his work, published his work, nobody can understand. Just a minute, I'll show you Graham Mendel's paper. Can you see? Hello students, can you see? Hello students, can you see this paper?
Yes, sir. Yes, sir.
Actually, this is Graham Mendel's paper, Experiments in Plant Hybridization. He published it in 1865. Okay, Graham Mendel. In his name, it is published. He actually read this paper. He has written and read this paper on 2 days, 2 meetings, like February 8th and March 8th, like in Brno National History Society, means Natural History Society. In this conference, Graham Mendel explained his work, but he did in different language. But one person called William Batson. You know who is William Batson? Who is William Batson? He is the one who coined the term genetics. Okay, genetics is the term coined by William Batson, and a strongest devotee or disciple or follower of Graham Mendel, inspired by Graham Mendel a lot. Okay, William Batson, the very famous genetist of 20th century. Graham Mendel is the 90th century scientist. He is a 20th century scientist. In 1901 only, he written this Grahor Mendeloda paper is translated into English actually. See, when you look into this paper, okay, when you look into this paper, it is very very difficult to understand because it is simple plant hybridization experiments. This paper tells about selection of the experimental plants, like why he selected that plant, like differentiating. See, he is explaining contrasting, differentiating character, okay, like all the things which we saw, why he selected this plant. So he explained, and then he performed experiments. Okay, he performed a set of experiments like seeds collection, all these things he was explaining, different in color, different in seed coat, okay, different in pod, different in like flower position, difference in long stem order health. So 7 characters as we saw, these are all the 7 characters selected by Graham Mendel. Okay, 7 characters selected by Graham Mendel. Okay, and then he also talks about these terms, like very important terms, dominance and recessive. Okay, scientific terms. He did not talk about genes or whatever, he just uses these terms: dominant genes, recessive genes. Okay, dominant gene, recessive gene. And then like the 7 characters and 7 set of experiments. Okay, first generation experiments, like form of seed. Okay, round seed, wrinkled seed. Okay, the shape of the seed. Okay, look at that. He crossed the plants, 253 plants he crossed and collected the seeds. 7324 seeds he collected, and what he is presenting, and then in that, out of that 7300 seeds, 5474 seeds were round, 1850 seeds were wrinkled, and then the ratio he gave 2.96 means 3 is to 1, that similar ratio. And similarly like that for all characters. Okay, out of the 7, all 7 characters, he got the results, like seed coat violet or whatever, so that color. And then like for all 7 characters he got the same 2.8, 3.1 means 3 is to 1 ratio, 3 is to 1 ratio. So like that for all the 7 characters he got. So that basically he published. And then look at that. No, in this paper you cannot see any other big informations. All are statistical, all are explanatory. No loss, no genes, no word denotes genes, nothing is present. Okay, so that's why people, just the important thing in this particular paper is the ratios. Okay, when you come to the presentation here, you can see the ratios. Can you see the ratio? Genotypic ratio F2, 1 is to 2 is to 1. Phenotypic ratio F2, 3 is to 1. Very very important thing. Monohybrid cross means you should remember the ratios, genotypic ratio and phenotypic ratio. So genotypic ratio is 1 is to 2 is to 1. What is 1? 1 homozygous tall, here 2 heterozygous tall, and then 1 homozygous dwarf. So this is the genotype, and the pure tall, hybrid tall, they are the phenotype. Okay, so in phenotype 3 tall, 1 dwarf. See why I am telling all these things. When Graham Mendel explained his genetic principles, actually nobody can able to understand his work. Okay, like it was not taken as the highlighted experiment. So Mendel was not happy with that. So people cannot understand genetics with Mendel's paper. But later, I will show you one thing. Can you see this presentation? Hello, students? Hello, students? Hello?
Yes, sir.
Ah, yeah. Later, Graham Mendel actually died in 1865. He published his work and died in 1884. So later, these are all the important people. Okay, very important, significant people. Ronald Fisher, Hugo de Vries, Cal Corens, Eric Teschmack, and then the other person, like I told you, the William Batson, and then Johansson, and then this person, Lion S. Magner. Okay, Laios and Magner. So these are all the people who reworked on Mendel's experiments. Okay, why the contemporaries, when Mendel explained, the contemporaries do not understand Mendel's work. Of course, they dismissed his work. No, we cannot understand it. It is simple number and ratios. We cannot understand your what is it 3 is to 1 or what is 2 is to something 961. They cannot understand these ratios. They cannot understand the pattern of inheritance. But later, these people, Reginald Fisher, and then say they actually worked in different groups. They are different people from different aspect they were working. Finally, like they also, when they did their experiments, for example, in 1890s, Hugo de Vries, okay, Hugo de Vries found that 3 is to 1 ratio again. 3 is to 1. Like he experimented on 15 different species and found the same ratio 3 is to 1. Okay, Hugo de Vries. And similarly, like that, other people also able to find what Mendel was got in his results. Okay, so together, all of these people, especially Hugo de Vries, Cal Corens, and Eric Teschmack. So these people only, like with their understanding, and in this period, lot of genetic terms were there in practice, like genes, genetics, genotype, phenotype. Mendel did not use the term genotype, phenotype, but it's a later period. So these people introduced those terms, and then they published Mendel's work as what? Mendelian laws or Mendelism. Okay, Corens, Teschmark, and Hugo de Vries published Mendel's concept, and Mendel, based on Mendelian experiments, they published his work on Mendel's laws or laws of inheritance. Okay, so actually speaking, Graham Mendel did not propose Mendelian laws. Okay, he did his work on these plants and published the ratios, F2 ratios of all 7 characters, monohybrid cross ratios, and in another paper, dihybrid cross ratios, 9 is to 3 is to 3 is to 1. That monohybrid and dihybrid cross we will be seeing in next class. But this is what they actually proposed. Means Graham Mendel published. But later, after the rediscovery of Mendelian principle by the later day scientists in 19th century, means 20th century scientists, and then after 35 years of break, Mendel published in 1865, and after 1900s only, these after 35 years only, the other people rediscovered Mendelian principles. Okay, so that's the most important thing in Mendel's experiments. So, like, even though people rejected Mendel's work, and Mendel was telling the community that my time will come. Okay, Mendel சொன்னாரு ஒரு நாள் என்னோட நேரம் வரும், இன்னைக்கு உங்க யாருக்குமே நான் சொல்றது புரியல, but one day you will understand my ideas and views. But definitely it happened after 35 years. So he is the first person to experimentally prove the pattern of inheritance. We call Graham Mendel as what? The father of genetics. Okay? Yes, with this, let me stop today's class and we'll go for a discussion. Okay? So, now tell me what and all we discussed. Hello, students.
Yes.
Yes.
We saw that Graham experiment on we saw Graham
Little more louder. Just a minute.
sir, can you sir?
Just a minute. Yes, Jishnu, tell me.
Graham Mendel's experiments on monographs. Saw why he chose plant.
I think you can increase your voice little louder. Yes, Jishnu. Are you telling something? No, Jishnu, I think I cannot hear. If others can hear, Sai Vignesh and Atish, can you hear properly?
No, sir.
No. Okay, okay. Jishnu, I think there is some problem with your mic. Okay, so let me ask from someone else. Atish,
Sir,
yes? Just like the things which we discussed in today's class.
there are different types of traits.
Okay,
Then different types of crosses.
different types of crosses. And then
Continuous variation, discontinuous variation.
hmm?
Continuous variation, discontinuous variation.
Variation, continuous variation and discontinuous variation. Very good.
There are two laws of monohybrid process.
Two laws connected with monohybrid cross. Law of what are the two laws?
Inheritance and the law of segregation.
Okay, law of dominance and law of segregation. Very good. Then Jishnu, you can type, no problem. You can type Jishnu. And okay, so let me ask you some, like me, like in flow, where we started and where we finished. So let me sum up today's class. Like we just started with what is genetics. So genetics is the study of inheritance and variation. And then I explained about, means I defined what is inheritance and what is variation? Inheritance means transmission of characters from parents to progeny, that is inheritance. And then variation means when the characters are inherited from parents to progeny, in what degree they vary from the parents. The reason of variation may be endogenous. Within the cell, some mechanisms can cause variation. And then exogenous factors. The factors from outside can cause the variation, but still there may be significant or insignificant variation. So, heredity and variation. That is the study of heredity and variation. That is genetics. And then also we discussed about three divisions of genetics. Why we need to study about Mendel's experiments in the genetics. So, to understand that, I just told you about three divisions of genetics. What are the three divisions? Classical genetics, molecular genetics, and transmission genetics. Classical genetics means it is the study of history and basic principles, how we understood mechanisms of genetic principles of genetics by classical experiments, like how Mendel did his experiments and what we understood from Mendel's experiment. So that is one thing. So classical genetics. And then molecular genetics. For the genetic mechanisms, what molecules are responsible? The DNA molecule, the RNA molecule, and the protein. DNA, RNA, and protein molecules. So they play a crucial role in determination of the genetic characters, the genes, the chromosome, the DNA, RNA, about their structure, form, formation. So that study is called what? Molecular genetics. And then population genetics, in the sense we will be like population, we are human population, some genes are there in my body, gives me a definite character, different character, like that you have a gene which gives you a different character. So in a populations, what are all the genes are common in nature, what are wild in nature, what are mutant type, what genes are mutated, what kind of another variant character is present in the organism. So like that, comparing the genes in organisms, okay, the characters in organism, that is population genetics. Okay, so classical genetics, molecular genetics, and population genetics. So to understand the classical, the basic concepts, we used Graham Mendel's experiments. Okay, Graham Mendel is monk. Okay, in his monastery, in a little garden, he cultivated a pea plant, the garden pea plant. And the important aspects of the garden pea plants are they are bisexual, easy self-pollination, easy cross-pollination, and more interestingly, all the seven characters are contrasting, very contrasting seven characters, because of what? That discontinuous variation. It exhibited the discontinuous variation. So the characters were contrasting to each other. So that we discussed. And then the life cycle, in few weeks we can see all the characters. If the seed is germinated, in another 3 weeks you can get your means in another 3 months, or I'm sorry, in another 12 weeks, yes, so 3 to 4 months you will be getting your new seeds. From the seed you will be getting, like, the plant will grow, and then you will be getting your seeds. You can analyze the character. So short lifespan, and then an easy cultivation method. Okay, so these are all the reasons helped Graham Mendel, made Graham Mendel to select this plant, Pisum sativum, pea plant. And then the seven characters we discussed, selected by Mendel: seed shape, seed color, pod shape, pod color, flower position, and then stem height. Okay, round wrinkled, yellow green, full constricted, green yellow, purple flower, white flower, axial flowering, terminal flowering, short plant, dwarf plant. So like that, contrasting characters existed in these plants. all 7 characters were selected by Mendel. And then the terms connected with genetics we saw. The terms are like genes, alleles, genotype, phenotype, homozygous condition, heterozygous condition, dominant gene, recessive gene. So these are all the things which we discussed. Okay, the terms connected to genetics with examples we saw. And then the crosses like monohybrid, dihybrid process. The first we just today, though there are different type of crosses, we started with monohybrid cross. Okay, monohybrid cross is nothing but a single character is selected and two different parents are selected, like one is homozygous for tall, another one is homozygous for dwarf. Okay, so two parents are selected and they are crossed. So the F1 generation all were tall, and in the again F1 progeny they were crossed, and then in the F2 progeny, like in 3 is to 1 ratio in monohybrid cross, most of the plant were tall, but still the dwarf plant was seen. Okay, this made like this is the main contribution of Graham Mendel. And like the important thing for you to remember is the monohybrid cross genotypic ratio is 1 is to 2 is to 1. The phenotypic ratio is 3 is to 1. Okay, in genotypic ratio, what is that 1 is to 2 is to 1? 1 heterozygous for tall, 2 sorry 1 homozygous for tall, 2 heterozygous for tall, and 1 homozygous for recessive. That is 1 is to 2 is to 1 ratio. And then though it is homozygous for tall or heterozygous for tall, it is the plant is seen as tall plant only. So 3 tall plant and 1 ratios of dwarf plant. Okay, so 3 is to 1. This ratio is the most significant finding of Graham Mendel. So almost 7 to 10, 25,000 I think. Okay, so 25,000 plants he cultivated, and then he recorded, he studied when the seed was collected, and again the life cycle, and when it produced seeds. So complete history of all 25,000 plants recorded by Graham Mendel. He kept it. He is the first person. Graham Mendel is the first person to apply maths and statistics to the science, especially in the plant hybridization experiment. That's why people said, "Veru numbers and ratio, we cannot understand anything. You go," like that. People said, but that is the most significant thing, which in the later part only we understood. So monohybrid cross, and because of this experiment, later the contemporaries may not accept Mendel's principles, but the later day scientists, especially Hugo de Vries, Carl Correns, and Van Teschmack, reworked on Mendel's principles, and then they said that yes, the pattern of inheritance, all the organisms, mostly the organisms, sexually reproducing organisms, for the inheritance pattern, they follow Mendelian principle. Hence, we call that area of genetics, if the organism follows Mendelian pattern of inheritance, they call it as Mendelism or Mendelian laws. Okay, the two important laws which we should know is law of dominance and law of segregation. Law of dominance says characters exist, characters are determined by factors, factors exist in pairs. In that pair, one is dominant, another one is recessive. What is dominant? The one which is expressed in the F1 generation is the dominant trait. The one which is expressed only in the absence of dominant gene, which is the recessive gene, that is seen in the F2 generation. Okay, so this is law of dominance. And then law of segregation. In law of segregation, we saw like it may be the dominant gene or it may be the recessive gene, but in meiosis they equally segregate, and then they have the equal chances for inheritance from parent to progeny through the formation of gametes. Okay, in parent organisms, dominant gene, recessive gene may be there, and this can segregate equally, and they can have the chances for inheritance equally to the next generation. So that's the most important thing about law of segregation. Okay, so Mendelian experiments, Mendel's characters, monohybrid cross, and then the laws of dominant, law of recessiveness, genotypic ratio of monohybrid cross, phenotypic ratio of monohybrid cross. So these are all the most important thing we should know. So rules of inheritance, in the sense law of dominance and law of segregation, are the most important aspect here we should understand from the first part of Mendelian experiments. Okay, so this is what we discussed in today's class. Yes, so keep this in mind and go through your book, and you can get a better idea also. So please read the book, and that is the thing mainly followed for your explanation. And if you cannot understand anything, please note down your questions. We can discuss this in person also, and in the next class we can continue with the dihybrid cross, maybe working out in offline, like in your board about the dihybrid cross will definitely help you. Fine. Anything else you want to ask? Yes, students? Okay, so please go through your material and note down your doubts, and please understand the terms first. If you cannot understand the terms of genetics, you cannot understand the principles of genetics. Okay, so please read the terms, get a better understanding of the terms, and then you can, if you read your book, then it is easy for us to understand genetics. Even you can help your friends to work out monohybrid cross, to work out dihybrid cross, to explain the principles of genetics. Okay, and thank you for your cooperation. Take care. Thank you, students. Thank you.